Hello, everyone, and welcome to today's workshop, Looking Back, Going Forward, Rethinking Genomic Data at HeLa 100. My name is Melanie Goisoff, and I am a research officer at BBMRI ERIC ELSI and lead of the Task Force Societal Issues. My colleague, Kaya Akius, has initiated today's workshop and also prepared the content. Before we start, I want to make you aware that the first part of the workshop, including a presentation and question and answer session, is being recorded. We will make the recording available on our website in the ELSI knowledge base. The second part, the discussion, will not be recorded. This workshop is supported by CY Biobank and BBMRI ERIC ELSI Services and Research. Special thanks goes to our colleagues named here for the fantastic collaboration in working on some of the key issues that are being discussed today. I'd like to introduce to you today's presenter, Kaya Akius. Kaya is a member of the BBMRI ERIC ELSI Services and Research team. He completed his bachelor and master studies in molecular biology and genetics and is currently a PhD candidate at the Science and Technology Studies Department at the University of Vienna. In the same department, he has been teaching part-time. So Kaya and I are currently co-teaching a course on knowledge production and societal aspects in postgenomics, and the idea for this workshop was born there. Kaya's interests range from non-health related genomics research to secondary use of genomic data, as well as science communication. A recent publication includes an article on how genes are associated with the social in specific data practices, which is mentioned here. The workshop will start with a presentation. I want to hand over to Kaya.
Thank you for the kind introduction, Melanie. I will start my presentation right away. But before doing so, I want to just tell you that we will be focusing on Henrietta Lacks first, and then the HeLa cell line a little bit, and then take a look at a 2013 controversy surrounding the HeLa cell line, where the HeLa cell line was sequenced for the first time. So I will just share my screen. I hope you can see it in full screen right now. HeLa cell line is probably the most important cell line in scientific history. But first, let's consider the origin of these cells and the beginning of the story. Henrietta Lacks was born as Loretta Pleasant in Roanoke, Virginia, on August 1st, 1920. This year marks the 100th birth year of Henrietta Lacks, and we will touch upon this point again. This famous picture of her identifies her as a joyful, self-confident African-American woman who has contributed not knowingly to some of the greatest developments in biomedical research. Her story contributed also to discussions of ethical, legal, societal issues within and beyond biomedical research. Following her mother's death at an early age, Henrietta was raised in Clover, Virginia, with her grandfather, as you see here in the picture, where the Lacks family was continuing to work the tobacco fields since the times of slavery. Henrietta married her cousin David Lacks in 1941, and they moved to Baltimore, Maryland, where there were opportunities for difficult work at steel factories for African-American men during the wartime. It was in Baltimore that Henrietta was diagnosed with cervical cancer at Johns Hopkins, one of the best hospitals still in the United States. But a biopsy that was taken from her was provided without her knowledge and without her consent to a researcher at Johns Hopkins, George Guy. So, George and Margaret Guy were working on developing a human cell line. Immortalizing human cells would have contributed a lot to science and medicine, and this would allow keeping cells indefinitely, producing high quantities, allowing work on the same line at different locations around the globe. So when the biopsy sample from Henrietta Lacks came, the Guy pair had, for the first time, the success of producing the first human cell line in 1951. Named HeLa for short, the cell line traveled around the globe and was shared with researchers without financial gain for the George and Margaret Guy pair, or the Johns Hopkins. But during the same time, a biotechnology industry started to form around HeLa cells, from culture media to disposables. While the cells were used for research, the first success stories started to attract attention in public. Here you see Jonas Salk vaccinating a child against polio. Salk's group developed the polio vaccine using the HeLa cell line. In the end of 1960s and early 1970s, it became more and more clear that HeLa cells were traveling not from lab to lab around the globe, being shared among scientists. They were also highly efficient in contaminating other cell cultures. It is now known that at least 32,000 research articles have reported research on cells misidentified due to contamination issue with HeLa. In 1970s, scientists identified genetic markers that would distinguish HeLa cells from others. This was possible only by contacting Lacks' family and drawing blood from the family members who did not know until then about HeLa cells, let alone that they came from Henrietta Lacks. But already in 1976, the name Henrietta Lacks was known to the general public. Rolling Stone magazine even featured a piece where the Lacks family was interviewed. The piece, titled "The Double-Edged Helix," started with the sentence, "In the delicate realm of biomedical research, those vigorous cervical cells from Baltimore are creating confusion and possibly disaster." Contamination story was taking over the success of HeLa in media. It is important to note that the cells were originally reported to be coming from a fictitious white American woman named Helen Lane, but genetic markers for African ancestry were already identified in 1960s for HeLa cell line, causing confusion. Finally, the obituary for George Guy in 1971 marked Henrietta Lacks as the origin of the cells once and for all. It is difficult to separate the story of Henrietta Lacks and the Lacks family from the story of HeLa cell line. For the sake of time, we will leave the general history at this point, but we would like to highlight the book, The Immortal Life of Henrietta Lacks by Rebecca Skloot, and the documentary with the same title for those who are interested. Since we are an international crowd, we would like to highlight also that the book was an instant bestseller that has been translated into numerous languages, from Greek to Turkish, German to Hebrew. Now we are moving on to the topic of today's discussion, an episode of HeLa controversy from 2013. And we have chosen the subtitle, The Chronicle of a Controversy Foretold, inspired by Gabriel Garcia Marquez's book with a similar title. Marquez discusses a death that is foretold, but the most striking aspect of the book is it was foretold because every actor in the book already knew this was coming. We also should have known the HeLa 2.0 controversy was coming. All the signals were already there in 2000s, 2010s. So what happened in 2013? First, a paper by a research group at the prestigious institution European Molecular Biology Laboratories, or EMBL for short, came out in the journal Genes, Genomes, Genetics. The paper by Landry and others provided sequencing and transcriptomics data for the HeLa cell line. The research made it to news on Nature and other platforms as an important development. After all, scientists had access to many genome sequences, including genomes of the fruit fly Drosophila melanogaster, the worm C. elegans, and many other species that were used for research. The news of HeLa genome was only a celebration as an important resource, but there was one problem. The origin of HeLa was known, even the family members and the pedigree was known, not to a few scientists, but to anyone who would search on Google or buy Rebecca Skloot's book. So on March 19th, EMBL sent out a press release stating that genomic data is not available due to privacy concerns of the relatives of Henrietta Lacks. On March 26th, Genetics Society of America, which is the publisher of the journal G3, also announced that the paper will stay online, but data will not be available until the issue is resolved. How did this happen? Let's take a closer look. When the paper was published on March the 11th, EMBL published a press release, as you see in the picture. HeLa genome is marked as an important milestone. The text had a number of questions, but one stands out as very relevant. EMBL asks, can we infer anything about Henrietta Lacks or her descendants from this sequencing? And the answer starts with, no, we cannot infer anything about Henrietta Lacks' genome or her descendants from the data generated in the study. Not everybody agrees on that point. One of the most well-known bioinformatics scholars, Yannick, tweeted, nice lie, EMBL, and he wasn't the only one. Professor of biology, Jonathan Eisen, as you see here, wrote a bit stunned that the people publishing the HeLa genome appear to not have gotten consent from the family. Besides social media, Rebecca Skloot's opinion piece in New York Times highlighted issues that there is much to infer from the sequence. Scientists uploaded HeLa genome to Snippedia, a platform for genetic analysis, and shared the results with Skloot. Skloot writes, no one knows what we may someday learn about Lacks's great-grandchildren from her genome. We know this. The view we have today of genomes is like a world map, but Google Street View is coming very soon. Looking back at this episode, one of Henrietta Lacks' grandchildren, David Lacks, says, we could leave it out there as is for the whole world to see, but the issue with that is when you sequence Henrietta Lacks' genome, you also include family trace of our genome as well. We don't know what would be known 20 years from now on, with the sequence just being out there for anybody to use and how that would have an effect on us. While geneticists and bioinformaticians, the family and the media reaction focused on what issues may emerge from the release of the HeLa genome to the open space, others claimed it was already there. One basic resource for molecular biologists and geneticists is the NIH's ENCODE, or the Encyclopedia of DNA Elements. And here you see a blog post that analyzed the sequencing data on ENCODE for HeLa, suggesting certain ancestry and disease risk information was already out there before the Landry paper. In short time, EMBL made a change to the press release, as you see. The colorful parts here are the ones that stayed the same. The most striking difference is to the question, can we infer anything, which has become, can we infer anything new? And the answer this time does not start with no. Over the next couple of months, NIH Director Francis Collins and the Lacks family, as well as Rebecca Skloot, met a number of times, and they have reached a conclusion that was announced in August 2013. Family consented to the sharing of HeLa genomic data with a restricted access model. The agreement created an advisory committee to the director of the NIH with the title HeLa Genome Data Access Working Group, and the database dbGaP was used as the access point. Researchers around the globe have already applied for authorization by submitting a short proposal for accessing the HeLa genome. There are 83 authorized accesses as of today. The agreement also applies to another publication on HeLa genome that was already accepted by Nature at the beginning of the controversy. You see the paper on the right. This paper by a University of Washington group was published exactly when the agreement was announced by the NIH. When the agreement was announced, Katie Hudson and Francis Collins marked the official stance of the NIH that HeLa 2.0 cannot set a precedent for future. They wrote, "It is important to note, however, that we are responding to an extraordinary situation here, not setting a precedent for research with previously stored de-identified specimens." The HeLa 2.0 shows that 2 papers by prestigious institutions and scientists submitted to prestigious journals were accepted without any concerns by funders, colleagues, reviewers, editors, even other scientists who read the papers. The question is, Is this a systematic issue that became foregrounded due to the publicity HeLa cell line generated, or rather an extraordinary case as Hudson and Collins claim? Let's keep this for the discussion, but going back to Skloot's New York Times piece, this episode highlights the following. The public supports the science and wants to help it move forward, but that support is dependent on consent and trust. Let's look back and try to go forward by doing that. We have provided you with a specific episode from 2013 and a bit of history of Henrietta Lacks, the Lacks family, and the HeLa cell line. For those who are interested, there is a great resource to look into further, and that is the HeLa 100 consortium. The consortium led by Lacks family has a number of events being organized in honoring the 100th birth year of Henrietta Lacks. Our activity is not under HeLa 100, but we certainly are amazed by the momentum generated with the effort. Now let's go back to history one last time and remember the discussions of practices that we today take for granted. In 1966, a groundbreaking paper was published in the New England Journal of Medicine by Henry Beecher. Titled Ethics and Clinical Research, Beecher identified what he regarded as ethical issues in top research of his time. He gave a number of examples, one of which was the following. I quote, "Example 17. Live cancer cells were injected into 22 human subjects as part of a study of immunity to cancer. According to a recent review, the subjects, hospitalized patients, were merely told they would be receiving some cells. The word cancer was entirely omitted." Beecher did not give the references and identities of the researchers who have conducted all this controversial research to protect the researchers from litigation, but these were provided to the New England Journal of Medicine reviewers. So they are true research that has taken place and published. In 1966, we find a legal case in the journal Science titled Human Experimentation, New York Verdict Affirms Patients' Rights. We realize this is one of the cases that Beecher discussed. Patients at a Jewish hospital in New York being injected live HeLa cells to test immunity against cancer 2 decades after the Second World War and Nuremberg Code. The verdict and the news in Science stress the importance of informed consent by showing what belongs and what cannot be regarded as consent and informing. What is remarkable is a commentary on this piece in the next issue of Science. Preston Burnham claims researchers are being pushed into a corner with the informed consent practice and claims, when we are prevented from attempting seemingly innocuous studies of cancer behavior in humans, as reported in the article, we may mark 1966 as the year in which all medical progress ceased. Did it cease? That is a question we may think of in the discussion. Now, back to today. We have incredible amounts of genomic data at our disposal as scientists, but also as ordinary individuals. It has been shown with numerous examples that it is very difficult to protect genetic privacy, as the genomic data itself is not at the hands or control of an individual. It is shared by relatives who also happen to share a number of identifiers like last name. And if any relative of ours makes their genomic data available along with other identifiers, it is no longer possible for existing anonymous data of ours to remain anonymous. For those interested, here are 2 great pieces to read. I will try to show you with an easier example that has made it into the news. In 2018, you might have heard a serial killer dubbed Golden State Killer was identified and became known worldwide. This individual, who was able to escape prosecution for years, rather decades, did not have his DNA on any forensic databases. He did not have his genome in any repository, but there were biological material from crime scenes that were stored. What the US justice system allowed was to extract genomic data of the Golden State Killer from crime scene material and upload this on a database named GEDmatch. GEDmatch is not a forensic database. It is not a scientific database. GEDmatch is a portal where customers of direct-to-consumer genomics companies can upload their genomic data to compare their genome to other participants on this platform. But how could the golden state killer be identified through such a system? Imagine the right-hand side of the figure to be the current population in the US, and the non-black figures to be on the GEDmatch platform. When a new profile is uploaded to such a system, for example, Golden State Killer's genomic data, the system automatically looks for the closest relatives by identifying portions of the genome that are shared. These are shared because the two individuals have recent common ancestors, for example, a common grandmother or a common grandfather. Once the closest relatives for the individual were identified, the police used the information to search for the Golden State Killer and at the end verified his identity by taking a sample, possibly from a coffee cup or a doorknob that he touched. This is not the first case, and it will not be the last. People often think of forensics when the topic is identifiability, but as the HeLa case shows, there's more to genomic identifiability concerns, from insurance to stigmatization. We would like to end the presentation with a few ideas that may be relevant for the discussion that will follow. A US Presidential Committee for Bioethical Issues report says in 2012, already before the HeLa 2.0, the following: because whole genome sequence data provide important insights into the medical and related life prospects of individuals as well as their relatives, who most likely did not consent to the sequencing procedure, these privacy concerns extend beyond those of individual participating in the whole genome sequencing. But HeLa 2.0 makes us ask whether non-identifiability is an illusion, or whether it is still possible to aspire for non-identifiability. This brings us to the question, even if there were to be a legal framework in place that would dictate asking relatives for their consent on genomic data, then the bigger uncertainty emerges. How far in the family should one ask for consent? How far in the family can one ask for consent? Such ideas necessitate reconceptualization of what we take for granted or thinking outside of the box. For example, similar concerns emerge with incidental findings and how and what to communicate to the family members, and how to align this with the right not to know or even right not to be known. With that, I would like to thank you all for listening to the presentation part of the workshop.